Alrighty. I say alrighty a lot, I think. I think it's just my way of getting started. It's really cold. So if I'm like, that's why. And the drop in temperature is kind of mucking about with my lungs. So I've stalled starting this because I just keep having coughing fits. So if I start coughing, my apologies. I just didn't want to wait any longer. Now, I am going to put both of these into one video because they're not super long. It's not going to be quite as last week when I slammed them all together, but I am doing both chapter 29 and 30 as one. A lot of this one, chapter 29, we already covered in class on Monday. I talked about it, but then that quick part of the human senses lab that we were able to conduct, I talked about a bit of this. So there you go. I think most of you already know the fact that certain animals have other abilities that humans do not. You've heard that about the dogs hearing a high-pitched sound. You hopefully have or remember in the book previously about bees seeing certain ultraviolet patterns in the flowers. What you may or may not know is about sharks. It's what I think is the coolest, where they can detect changes in the electric field. And I've watched, I cannot tell you how many documentaries on that. I think it's fascinating. And then migratory birds, fishes, and turtles able to navigate the Earth's magnetic field, and that's how they're able to do their migrations and stuff like that. So a lot of different senses. So there you go. Pretty turtle. Sensory reception, hearing and balance, vision, and taste and smell. And I'm so glad I talked about Colin in class on Monday, because when I cover this, the hearing and balance, you're going to hear me talk a lot about him. And I just realized I should go ahead and turn on that laser pointer so I don't go, oh, wait a minute, I should turn this on. I already have it. So sensory reception. We have various sensory receptors throughout our body that help us with the sensory information. So we have the sensory transduction that's going to convert the stimulus energy into the receptor potentials, which causes the entire chain reaction, which goes to the brain and then throughout. So this ties into the last chapter we covered, showing you here that this girl's licking an ice cream cone. So not the cone, she's licking the ice cream on the cone. So by licking that, it's activating the taste bud. The taste bud has the sensory. This is actually a chemoreceptor, just so you know. And then that causes the chain reaction that then goes to the brain. The brain detects something, which flips a switch that does more. So this one is because, like this, then the tongue tastes it, sends the reaction to the brain that says, hey, we've got a buttload of sugar, and then the brain goes boop, and then kicks out the stuff to help break down the sugar. So there you go. Okay, we talked about action potential on Monday. This is just continuous of that, where it is action potential frequency reflects stimulus strength. So the stronger the stimulus, the more action that takes place. That's pretty much it. And then we have something called sensory adaptation. When we do the full human senses lab, I actually cover this when I'm talking about the mechanoreceptors. Mechanoreceptors, think mechanics. So that's things you touch. And in the human senses lab, when we can conduct it, I always use the example of glasses, or like when you get dressed. Glasses I love to use because it's one you can test out and play with in the class. Whether or not you got dressed, not really a good thing to test out in the class with people, but normally when I teach the lab in class, I look around the room and I say, I can tell everybody in here got dressed this morning. Mainly because if you didn't, you wouldn't be in the classroom. But when you first put on your shirt, you normally feel it when you first put it on, but then after a while, you don't notice it anymore. That's because your body gets used to feeling it. Glasses. You can sit here like this with no glasses, feels normal. You put them on, when you first put them on, you totally feel it here, you feel it here. Then, after a while, your body gets used to it, you don't even notice it anymore, and then you'll see the people walking around looking for their glasses while they're wearing them. The other one I like to use, because you may have seen me play with it a lot, rings. My rings are a little loose, and so I play with them to make noise. It all depends on my water retention, basically. And it's cold, and I've been drinking a lot of water, so right now they're very loose. I could actually move them to another finger, but because I can do this, they're constantly moving, so I am always noticing it. This ring, however, even though I can move it too, it's only one ring, it doesn't move as much, so I don't notice it. So that again, sensory, you get used to it. There you go. All right, so now this is showing you the taste bud and the sensory neurons, and we have no sugar. Then as it gets sweeter, we have more action happening. Same thing for no salt, more salt, action. But if you keep the same amount constantly, it kind of gets used to it and just, doesn't really activate anymore. It's actually how a lot of addictions work, like nicotine and stuff, just so you know. So what is meant by sensory transduction? Oh, there you go. We already covered it. It's where you're taking the stimulus, turning it into an electrical signal by the receptor. There you go. Now, here, I do think this is interesting, where we haven't quite figured out exactly how these animals are able to sense the Earth's magnetic field, but there's no way to deny that they do, because they follow the same paths. And the magnetic field in the past has reversed, and if you feel like looking it up, there are some current articles out right now as they're detecting the shift in the magnetic field again. So, really interesting, but we don't know how they do it. We just kind of think maybe kind of thing. So, that's what this is about, saying that they have different types of hypotheses, suggesting it has to do with the receptor cells or biochemical reaction, different things like that, but we don't know. And it's okay not to know, because you can't know everything. Otherwise, gosh, life would be boring. And then, of course, you would stop looking for answers, and then you would never know if you were actually wrong. So I guess it's a good thing we don't know it all. Anyway, here is a way that they're trying to find out if these magnetite-containing cells, there you go. So they have the cell suspended in fluid, subjected to rotating magnetic field. So it's going round and, well, I guess it's more like that for the picture. And then a few cells spin in response to the rotating magnetic field. So if some of the cells are spinning in response to the rotating magnetic field, it wants to know, do the cells then contain magnetite? If scientists remove the neurons essential to magnetoreception, I cannot get these words out, for roundworms, predict what will happen when they alter the magnetic field around the worms. So if you already know that these worms are reacting to a magnetic field, what happens when you take the thing away that recognizes it? Well, can't orient themselves. Not quite the same as, but the two things that I can give you as a sort of example, but it has nothing to do with the magnetic field, which is why it's not quite right. One, we are going to talk about in a little bit, like when you spin around and then, you close your eyes and you spin and then you're like that and you try to orient yourself. Kind of the same thing, because they're reading the magnetic field to know where they are. When you're spinning around with your eyes closed, you're kind of removing yourself from that. Now the reality is it's the sloshing happening in your ears, and that's why I said we're going to get to it later. That's really what causes the and then you don't know where you are. But it's same kind of thing where you're removing the part that helps them orient. The other one I was going to say has to do with ants, because if you've ever watched that long trail of ants, normally you see one or two ants and then they go back exactly the same way and then they all come that way. It's because they're letting off these little pheromones, laying down a tiny little trail that all the others follow. If as one ant goes here, you take your finger and you do a circle around them, and do another one, just do a couple like that, like, you know, smaller, larger, larger. You break that little field that he did, and then you'll see that sucker will just be walking around in the circles that you created because he can't figure out where to go because you removed the part that he senses. So same kind of thing, but this is magnetic. What I was talking about is not magnetic. I hope that didn't just get confusing. I'm so sorry if it did, but my brain is like going crazy today, so sorry. Not the best day to do videos, but your test is coming up, so I have to do the videos. Anyway, well, actually, I guess I technically don't have to. I think I'm the only one at GMC doing all these videos. But I don't know. If I was taking the class, I would want more than just the book. So there you go. Anyway, these are the things I sort of talked about in class on Monday. I've made reference to it. I've already talked about the mechanoreceptors a second ago. So here you go. We have thermoreceptors. Thermo, think of thermometer. These are the ones that are going to detect whether something is hot or cold. So like if you touch something with your hand, like, you know, reaching towards the stovetop, like, whoo, that's hot. Those are your thermoreceptors that got triggered. Mechanoreceptors, mechanical energy, touch. So that is your mechanoreceptors. Same thing as when you, like I said, put on a shirt, put on your glasses, that kind of thing. Pain receptors. These are the ones that, in my personal opinion, are very, very interesting because how different they are for different people, because you hear about different people having different pain tolerances. Some of it really is a tolerance level. Some of it has to do with how well the pain receptors work. The pain receptors are there to help keep you from being in as much danger, because if your body hurts, it's letting you know, don't do that, for the most part. But then you have these crazy disorders where people don't feel the pain. My mom is on that end. It's not that she doesn't feel any, it's just she really does not feel most of the pain. So she'll think something is, you know, perfectly fine, and then she goes to the doctor. Like 2 weeks ago, I took her to the dentist. She goes in there and finds out that she has a hole all the way in her tooth and needs a root canal because the whole thing's gone. And the dentist is like, don't you feel pain? No, she truly didn't notice anything. She has a lot of doctors who have to keep an eye on her because normally, clearly, somebody will say this hurts or that hurts, and she doesn't notice at all. So it's really interesting. But then this is also where fibromyalgia seems to be jumped in, where it's like those suckers are poof, amplified. So there are people who, like my mom, they don't seem to work. But then on the flip side, there are people where they are like overstimulated. A breeze can go by and it triggers the pain receptors. And so don't make fun of somebody if somebody says something hurts. Listen to them. You don't know what that person's feeling, basically. So there you go. Anyway, tangent over. Chemoreceptors. I made a comment about this a second ago when I was talking about the ice cream. Chemoreceptor responds to chemicals. That's what's happening when you're tasting something. It's literally a chemical reaction. When I do the lab, I also normally talk about that's why you don't mess with somebody with something hot. If you know someone who says they can't handle spicy food, it's not funny or even remotely nice or okay to purposely give them something hot so you can laugh at their reaction. They can literally burn their mouth. It can cause a chemical burn in their mouth. This is also the reason why when some people, and most people know at least one person who has done this, punish their kid with hot sauce to the tongue, because that's something some people do. That's actually child abuse, because the child's mouth is not designed to withstand that chemical. It is no better than if you literally dropped hydrochloric acid on their arm. So what's happening in here is actually a chemical reaction. So don't be mean, all right? My tolerance for spice is like here. I love spicy food. My mom's tolerance, way down here. She can't handle it. She thinks black pepper is spicy. You should see when I order her food, I actually type in there that bland is good. She can't stand spices. And I even put in there she thinks black pepper is spicy. Meanwhile, I am the person who is adding like everything because I like good spice. So, there you go. Different strokes for different folks, right? And then we have the electromagnetic receptor, which responds to electricity, magnetism, or light. In class Monday, I talked about the photoreceptors, which is the light receptors, because photo is for light. That's why photosynthesis is synthesizing light. So there we go. Click. Sensory receptors in human skin. I almost got rid of this slide, but I decided to keep it because I wanted to show you this. So the epidermis is the top part of your skin. What is below that is the dermis. Somebody touches the top part of your skin, you can feel it. A lot of times it's because it hits the hair. The hair-like structure gets triggered and it causes more feeling down here, for lack of a better phrase. Notice here, pain is one of the highest parts. Light touch, you need a little bit more. Heat and cold. Cold is a tiny bit higher than heat, not by much, but all but pain seems to be down in the dermis. It's a little farther down. Epidermis, again, that's the top layer. So, yeah, I think that's good enough. Some people can't stand snakes. I think they're fascinating. I actually studied them in grad school. So, this is just showing you the heat sensing pit organ in a snake. So that's not their nose, it's not a nostril, that's actually its own organ that is searching for heat, because that's, what they do. Mechanoreception by a hair cell, just again showing you that the hair cells, it's not actually hair that they're showing here. It's the little pieces that react. It feels it. It's on the receptor cell, so it gets triggered, which causes that. This is showing you they're normally like this. Then there's a movement that goes this way that causes a reaction, and then this way that chills it back out. So there you go. Sharkies, because we know I love my sharkies. The lateral line is the line that goes down their side. Normally we would have covered fish and sharks and stuff in class, and when that happens I normally point out the lateral line because I think it's really interesting and it's how they like sense everything in the water. It's the reason why if you've ever tried to scoop up a fish like that, like a bear does, and I don't know how many people have ever tried that. I of course tried it because why wouldn't I? But you sit there and you think you can do it and you try to sneak up on it. As soon as you do that into the water, the fish takes off. It's because you hit the water current touched the lateral line, which let it know you were there. The lateral line is also how the schools of fish know to stay together, because as they go this way, they feel the shift and they all just follow. So I think that's pretty cool. Nares, all of this is involved in that whole electric part for the sharkies. I think they should show a hammerhead because they have even more across here, the Jacobson organ. So anyway, for each of the following senses in humans, identify the type of receptor: seeing, tasting, hearing, smelling. There we go. Already covered the photoreceptor for seeing. We've already covered the chemoreceptor for tasting. We already covered the mechanoreceptor for hearing. We did not cover chemoreceptor for smelling. That's another part we normally got to do in the human senses lab that we couldn't do this time. In the lab, if you look at the PowerPoint, it just says Skittles. What we do is you get to test that whole, if you hold your nose, it takes away the taste thing. And it really does work if you do it properly. If you do it properly, it is you pinch the nose closed completely, you put whatever in your mouth, you chew it up, and you swallow, holding the nose totally closed the entire time. You will not notice the scent, the taste, until you let go. Now, when people go, oh, I've tried that, it doesn't work, it's because you let go too soon. So that's why I use Skittles normally in this lab, because it's a very powerful flavor, but you could totally do this at home. This is also the reason why when people smoke, if you've ever heard that it deadens some of your taste buds, it's because you're burning the hairs and stuff in your nose. It's affecting your scent capability, which affects your taste. It's why a lot of times when you're sick, things just don't taste the same. It's, again, clogged up. It's affecting your sense of smell, which affects your sense of taste. So there you go. Hearing and balance, what I was talking about a moment ago. So for our ear, we have the outer ear, the middle ear, and the inner ear. So here we have human ear channel sound waves through the outer ear to the eardrum, to a chain of bones in the middle ear, and to the fluid in the coiled cochlear, the inner ear. Pressure waves of the fluid bend the hair cells of the organ of Corti against a membrane, triggering nerve signals in the brain. And a bird just flew down and landed on that bush and disappeared, and that kind of totally distracted me. I think there's a chipmunk up in that little tree there. So yes, I am getting very distracted by the various things around me. So let me go into detail on these things. Here you go. Here's our picture. We've got the outer ear. That's what you think of when you think of your ear. Middle ear, that's where you get to the eardrum. Inner ear is all the way in. That's where the cochlear is. And there's that bird again. And it's, how do birds just hang on the outside of your house? And there's another bird. I wonder if they're doing, oh, and now one of my cats has noticed the bird. This might get very interesting very soon. Anyway, so this is the main part most people think of when they're thinking of the ear. This is your auditory canal, because this is where the sound is coming in. This is where you think of when you're thinking of hearing. This is your eardrum. Middle ear here. Cochlear, this is part of your hearing, and this is also the fluid-filled part. This is the part that works with balance, and like if you've ever noticed when you're rocking a baby, it soothes them. That has to do with the inner ear. That has to do with the fluid going. It's a very soothing thing. And then this tube. If you have ever known someone who they kept getting the same kind of swimmer's ear infections as a little kid, and then they said they had to get tubes in their ears, this is the part that they're replacing. When you have an ear infection, this is normally the area that's massively infected. Here is where a lot of that pain is coming from, and this is why if you have a bad ear infection, they tell you do not pop your ears, do not try to do that, thing that goes poof to pop them, because you can actually burst your eardrum and then you are screwed. So don't do that. All right. Just showing you a little bit deeper into here, showing you all the different parts, because these are actually little bones that work up to make all the sounds work. If you, and like seriously, I'm not going any deeper than this for hearing. Obviously, if you take A&P, you're going to go deeper. I just find it fascinating that you have this and these bones, and then this funky thing that looks like a snail that actually makes all the tiny little vibrations turn into all the different sounds that we hear. I find it fascinating. I want to say it was in the 80s. There was a movie called Inner Space, I think. It had Martin Short, David... crap, I already forgot his name, but it had Martin Short, Meg Ryan. Crap, I can see the guy in my head. He's got like one of the best smiles ever. He always looks really friendly, and he was in a movie recently where he played a bad guy, and it was very mind-boggling because you just don't. Dennis Quaid, I think that's his name. But anyway, in this movie, you actually get to see this character. Dennis Quaid is inside Martin Short, and he's driving around, and you get to see all the different body parts on the inside, and it was kind of cool. But anyway, I always think of that when I look at this stuff. I think the way the human body works is fascinating. I just never wanted to actually be part of the massive studying of it. Can't be a doctor. We already talked about this, not me. Anyway, the ear will convert the air pressure waves to the action potential, and then that is what we perceive as sound. Louder sounds generate more action potentials. Kind of makes sense, right? More of a bam, you're going to react more. pitches will stimulate different regions of the organ of Corti, and deafness can be caused by the inability of the ear to conduct the sounds, resulting from middle ear infections, a ruptured eardrum, or stiffening of the middle ear bones, which is an age-related problem. And deafness can also result from damage to sensory receptors or neurons. There's a lot of things that can cause you to go deaf. But first I want to point out the first two parts here. The louder sounds generating more action potential, and the pitches stimulate different regions. Okay, you've probably noticed some people can handle loud better than others. I don't do well with loud. I actually always have on me a set of earplugs to try to bring down the decibels around me. And we have a little thing that talks about sound levels in a moment. But the bigger one for me is the pitch. There are certain pitches that actually give me physical pain. And I've had a lot of people just sit there and say, oh, I just need to get used to it. But that's kind of like sitting there stabbing somebody and telling them they just need to get used to being stabbed. This goes back to the whole don't tell somebody to just get over something. That's kind of cruel. I thought it was interesting when I learned that a pitch stimulates different spots in this organ, and that would be why people respond differently. So I thought that was interesting anyway. And yes, Fang climbed up on me again. Well, climbed up here and was about to hit the computer, which is why I then grabbed him and moved him. So this is just trying to show you an idea of what is being triggered with the different sounds and how it goes. Yeah, the previous text actually had a table that broke down the different decibel levels so you could see the dangers that you ran into with different sounds. Because, as you know, a concert is very loud, and a lot of times if you've been at a concert or a club, you leave and you have that ringing in your ear and it feels almost like there's cotton in your ear. That's actually the first indicator of hearing loss damage, and you have to give your body time to heal so you can actually heal your ears. Yeah, I think I'll just stop on that because I'm going off on yet another tangent. So what causes bending of hair cells at different locations along the basilar membrane, and how do these differences affect perception? Well, different sound frequencies cause different regions to vibrate. The hair cells, again, little hairs, they bend at different locations. And what happens is the more they bend, that's affecting how well things work. So if you're hitting it so hard that they're like that and then they finally flatten out, and I used to have a picture of this, it was really fascinating to see it, but the too loud constantly causes them to bend and not bounce back up. Once they never heal again, that's permanent hearing loss. When they're wilted down because of the amount of pressure, but then they slowly start to come back up, that would be the temporary hearing loss. That would be like you went to a concert or a club, you left, you have that ringing in your ear and everything's kind of cloudy, and people are talking and you're doing the what? That's because these little hairs are kind of stuck down and they're working on coming back up. So I have something else I want to say, but I want to make sure I put it in the right spot because it has to do with hearing and the hair structures. So I'm going to go a tiny bit more, and then if I don't see it, because it's one of those things that when I was doing the PowerPoint, I went, I wonder if that's why, and I looked something up and I want to share it with you. So first we'll go here. Several organs in the inner ear allow you to sense movement, position, and balance. This is the fluid-filled part I was talking about, where like if you spin, you get dizzy, or when you're rocking the baby, it calms them. So that's what these are. All three of these structures operate on the same principle, which is the bending of hairs on hair cells. So the fluid is still doing the bending of the hair cells. This is showing you the fluid, so as you move, it's making it do that. This is how, if you close your eyes and you're like this, you can still tell you're sitting up. If your eyes are closed and then you lean over this way, you can tell you're leaning this way because it's causing all the fluid to, lean the way you're going. This is also what affects you when you're just standing there and then you feel like you're starting to fall. It's a fluid shift, that kind of thing. So, yeah. Here are those little hairs that we keep talking about that are being triggered. So what type of receptor cell is common to our senses of hearing and equilibrium? Well, what do you think that would be? Mechanoreceptors, yes, because it has to do with a type of touch. Okay, I'm going to mention this because I don't think it's going to get to, because I think this is the last part we do for the hearing. So, the hair-like structures, the little hairs on the hair cells in your ears, where my brain went with it was cancer. Because this is what, 2020? 2018 is when my mom's twin sister died of cancer. And it was like one of those that out of nowhere. As in, she had some issues, she went to the doctor, she got checked, she got a second opinion because the first person said there was nothing wrong and she knew something was off. She got the second opinion and was diagnosed with ovarian cancer. They did the full hysterectomy within a month, of her first appointment. And they searched for other stuff, and then they found that it had gotten to her lymph nodes. Then they did the aggressive chemotherapy, and it was, long story short, basically she died in less than a year from the first notice something wasn't right. But it was during the chemotherapy that I learned even more. We talked about all this stuff, and we talked about the hair loss and stuff like that. And so when I was doing this and I was thinking about the little hair-like structures in the ears, it got me thinking about it, and so I had to look it up. And yes, chemotherapy can cause hearing loss because it can cause those hair-like structures on your hair cells to curl up and die. Sometimes they get replaced and it ends up being a temporary hearing loss. Sometimes it permanently kills them and then you end up with permanent hearing loss. And I just thought that was interesting. I wouldn't say don't get chemotherapy if you have cancer because of that, because if that seems to be the best way to beat cancer, I'd rather beat cancer. But I just thought it was interesting. Anyway, yes, and that is how my brain works. When I'm going over these PowerPoints, I'm like, oh, I wonder, and then I look stuff up, which is why sometimes it takes me longer than it should to get to things, because I swear I think I live my entire life constantly coming up with more questions and having to find answers. Not a bad way to live my life though, right? So, here we go. Motion sickness. Most people know people who get motion sick. Most people have gotten some form of motion sickness before. So here we go. Motion sickness may be reduced by closing your eyes, focusing on a stable horizon, or taking something like Dramamine. So while this sits on the checkpoint question, I'm going to talk about this. If you've ever been on a boat, notice the smaller the boat, the more likely the motion sickness, and it's because you feel the motion even more, so it's causing this to happen in here while you're looking at stuff. This is why they tell you to try to look at something solid. Whenever I started to feel a little on a boat, I did that. I just closed my eyes, and I had people tell me, don't do that, it's going to make it worse, but it was one of those things that I just naturally did, and it helped me every single time. I've never had Dramamine, or however you pronounce it. I've been on a lot of boats, little ones, big ones, whatever. And normally it's only the little ones that really cause me an effect. And I think I just saw a stray kitty out there. Like you can even see what I'm seeing. It's just this is why I keep getting distracted. I'm like, ooh, what's that over there? What's that? What's that? But anyway, now for this checkpoint question, we're going to do this. I'm going to give you the answer, and then I'm going to give you another example, which is kind of dated, but you'll understand. So, explain how someone could suffer motion sickness when watching a film shot from the front of a roller coaster. So, in other words, you're just sitting there perfectly still, but you still get motion sick. It's because of the conflicting information. Your body, your ears, the fluid knows that you're perfectly stationary, but your eyes see it going crazy. So there you go. That's why it can happen. Now, the example I was going to use is the movie The Blair Witch Project. I don't know if anyone here has ever heard of it, looked it up, knows any details about it, but when it came out in theaters, it was hilarious. I really recommend looking up the news articles on this. So many people were getting sick because of the way they did the camera. It was horrible. And I have a pet peeve about it because, quite frankly, if the movie is supposed to be about film students, then somebody would know how to hold a freaking camera. Just gonna throw that out there. But anyway, there was so much jostling with the camera work that in theaters across the country, people were throwing up in the theater while watching the Blair Witch Project because it was causing motion sickness. I thought that was funny. I was pregnant. I think this was 99, and so that would have been with my second kid. But I just remember being pregnant, watching it, and I started to feel motion sick, and I very rarely get motion sick. I started to feel it, and so I just closed my eyes and I just listened to a chunk of the movie until my stomach settled, and then I'd open, watch a little bit, and close it. It was a little crazy. But on a smaller screen, it's not nearly as bad as on a movie screen. Vision. We talked about this a lot in class, so this is going to be the part we go fast. Already talked about our eyes are set up in different ways. We have here is a planarian. They just have eye cups, so this is how they notice different things. Their eyes do not work the same as ours. This is the one I find the most fascinating. This is a fly. Look at these eyes. It's so many tiny little itty bitty things that make up their eyes. Something that's more fascinating is if you want to look up the mantis shrimp, they have the most incredible eyes. They also have the widest range of what they can see. The mantis shrimp alone is just friggin' phenomenal. Totally look it up. This we covered on Monday in lab. So this is the eyeball. We covered all the parts, so I'm not going to cover it all again. It's just we already did this. So here for the checkpoint question is just what is the function of the lens? Well, function of the lens is to focus the light to the retina so then we can see stuff, because the retina is where our photoreceptors are located. Talked about the different ways to focus, and so there you go. We've already got it. Talked about this on Monday in class. We talked about being nearsighted versus farsighted. I said myopia and hyperopia. Here's the nearsighted, farsighted astigmatism. Here we go again. So this is where I talked about the focal point nearsighted ends before it gets to the retina. For the farsighted, it is past the retina, that kind of thing. Did not really cover this, so I'm going to talk about this real quick. You have cataract and you have glaucoma. Glaucoma I did talk about in class because when I was talking about the vitreous humor, I talked about glaucoma because I was talking about the pressure in the eye and when sometimes they have to literally remove some of the fluid. So that's the glaucoma one. Aqueous humor. Forgive me, I said vitreous humor a moment ago. Aqueous humor. But still, too much pressure in the eye, has to take care of it. The cataract, we have a picture here. There. Cataract is this little film that kind of grows over it, and it makes it where you can't really see as clearly, and they can totally work on this one. They can do surgery. My mom has actually had cataract surgery. And it's very interesting, very fascinating, and very disturbing all at once because my mom has some of the most intense blue eyes you will ever see on this planet. I will have to show you a picture at some point, but they are those very light piercing blue. It looks like she can look all the way through you and through the next couple of people behind you. They are so this particular shade of blue. she had cataracts, she had the cataract surgery, and now when I'm talking to her, if I look certain angles, I can actually see the circle where the cataract had been, so I can see like the outer part, which is really interesting. But anyway, the checkpoint question here is, how does it affect vision? Well, reduces the amount of light reaching the photoreceptors, so that causes blurred vision and faded colors, because again, the purpose of the lens is to focus the light to hit the retina at the back of your eye, which has all the photoreceptors. So if you put anything over that, it's going to affect the vision. We covered rods and cones in class. Did that at depth, so I just did this to remind you the cone means color. So if you can just remember CC, cone color, you'll be able to remember that the cones are how you see color. There you go. So cones are for light and for color. Rods are for when it's dark. That's pretty much it. Again, just reminding you, photoreceptors goes from light through the lens to the retina and hits the stuff, and that's what makes it where you can see. So explain why our night vision is mostly in shades of gray rather than in color, because it's your rods, not your cones. There you go. Taste and smell. Talked about a bunch of this earlier in the PowerPoint when I told you what we would have done. Actually, I just noticed one other thing it said here. Aha, 5 taste sensations. I wanted to point that out because we have sweet, we have sour, we have salt. Look at me freeze for a second. Sweet, sour, salt, bitter, and yes, I keep getting distracted by more things. I should not be doing this today. See me trying to remember it all? I'm like, okay, we've got the bitter, we've got the sweet, we've got the sour, we've got the salty, and I'm wondering if the 5th one their meaning is umami, which is savory. I think that might be it. Otherwise, I'm straight up forgetting one. But umami is the one I always spend time talking about in the lab because that is a more recent development. And they discovered it. It's the flavor of what you get, like that deeper flavor in soy sauce or steak, different things like that. It's very particular. And then I like to point out that America a few years back tried to stake a claim of another taste, and that was fat. They wanted to stake a claim on the taste fat. And a lot of people laughed about that. They're like, oh no, but that's qualified under this or that. And I would honestly agree that fat has a particular taste because I don't like it. And it causes issues because I really hate that taste. And yes, I don't use butter. I hate butter. I hate the flavor of butter. I don't like most fried foods. I am the person, if I get something fried, more often than not, I'm going to peel off all the breading because I don't like it. So it's just weird. Anyway, 5 taste sensations. And I already talked about how smell and taste go. So this is showing you smell something and it triggers stuff and it gets it going. I don't know if we'll go more into it later, but normally when we cover the brain and we're covering the brain parts, we actually talk about one part of the brain that will take your senses and blend it with your emotions. And it explains why sometimes you will smell something and it will trigger a feeling. So like if every time you went to grandma's house, there was a certain scent, but like, I don't know, this is going to sound so weird, I'm sorry, mothballs. That's what it always was for me. I would visit my grandparents and it was always a very calm and sort of safe feeling that I didn't have anywhere else. And one of the things was they would open up the chest and they would pull out a blanket and they had mothballs in there. Mothballs smell so funky. I do not like the smell of mothballs. But to this day, as soon as I smell it, I feel calmer. And I think it's because of I would sit on the couch or the bed or the chair. Normally it was the couch. And they would get that blanket and they would give it to me. And then I would take it and I would hold it like this and I could smell the mothballs. So for me, that would be it. Most people, it's something that was cooked. It's also, these being connected, part of why, and we covered this before when we talked chemistry for digestion, when you smell food cooking, all of a sudden the salivary glands start kicking in to be ready, because it's all connected. So I think it's interesting. And yes, I know I went on a tangent, but that time it all went together. So what is the key difference between the cellular structure and function of taste receptors and olfactory receptors? Well, taste receptors will release the neurotransmitters, which cause the action potentials carried by the brain to the brain by the sensory neurons. Olfactory receptors are already the sensory neurons. So taste receptors do this chain reaction. Olfactory receptors, that's already what they are. So there you go. I love this one, the cilantro thing. The previous text didn't cover this one, so I was very happy to see this here. A lot of people love cilantro. Then you have the small percent who can't stand it or are just meh. I hate it. To me, cilantro literally tastes like soap. The first few times I had it, I honestly thought somebody didn't wash the utensil properly or the dish properly. And then I finally learned about this, and I was like, huh, interesting. Turns out it's a genetic thing. They did some studies. They found out that there's actually a genetic proponent for this issue about cilantro. So while some people think it's wonderful and they think it's funny that people are like, oh, I don't want the cilantro, it's because you don't taste it the same. So I think it's supposed to be good for you. And gosh, what is it? The seed. Is it coriander seed, I think? I don't know. There are seeds and other herbs that are made from the seeds of the cilantro plant that I can actually use, but the cilantro leaves are what taste like soap to me. Bizarre, right? So what would it mean if identical twins were no more similar in their preferences for cilantro than fraternal twins? Well, then it would be environment, not genes, because identical twins have the same genes. Fraternal twins do not. I think we covered that in Bio 123, just so you know. Okay, so we covered central nervous system last time. We did central nervous system and the peripheral nervous system, so this is just bringing it back to that, pointing out that this is how everything works together. So sensory receptors send it to the brain, which sends it to the motor output, which causes you to react. So what three general types of neurons are involved when you hear a noise and turn your head? Well, sensory, inter, and motor. Totally covered this on Monday. I know I did. And then that brings us all up to the what we should now be able to do. We're going to go, there's going to be that little segue as I edit, and then we're going to go right on into the next chapter about how animals move. Alrighty, we're already back to how animals move, and I said alrighty again. One of these days, I swear I'm going to figure out how to stop doing that. Probably won't happen, and now that I've had this one thought run through my head, now I just keep thinking of Ace Ventura. But anyway, how animals move. A recently developed genetic test may help parents identify which sport is best suited to their child's potential. I say go by what the kid wants to do, but that's just me. This is just talking about muscles and the skeletal system, that kind of stuff. We already talked about some of the muscles previously. We're going to talk some about the movement and locomotion, the vertebrae, skeleton, and muscle contraction and movement, which again, we've talked some about already. So most animals are fully mobile. Notice it says most, not all. If you remember, sponges are animals. They don't move. There's a couple of different animals that don't really move, but most of them do. Locomotion is the active travel from place to place. You have to have energy in order to make this happen because you have to go against, well, gravity basically, because gravity's just kind of hanging you, holding you there. You have to expend energy to move. So Animals that swim are supported by water, but they are slowed by friction. If you have ever noticed how you move on land, like if you walk on land and then you get into a pool and you try to walk in there, it slows you down. It's the friction. So that's all that's talking about. Pretty much we're all having to overcome some form of either gravity, friction, or both. Yeah, there you go. Contrast swimming with walking in terms of the forces an animal must overcome to move. Well, there you go. We already said that friction resists the animal moving through the water, but the gravity doesn't have as much effect because of the buoyancy. This is why, like, you float, you bounce in the water. You can go woo, woo. I'm sure I'm not the only person who does that in the water. You do that quick little thing and you're bouncing off your toes and it feels like you've got that tiny moment of being super duper light. That's the buoyancy they're talking about. So gravity still works, but not nearly to the same degree, but you're having to go against the water, so friction. When you're out here moving around, there is no friction right here, but there is gravity. So that's all that's talking about. Nemo! Actually, it's not Nemo because, see, both of his fins are good, but it is a clownfish. And then we have a dog running at full speed, so you can see again difference between swimming, running, and then we have kangaroos hopping. They are so interesting. They can beat your butt too, man. Those suckers can be mean. Worm crawling. Okay, so the way this sucker works, and I purposely keep this in, I cover it every quarter that I do Bio 124 because I find it so interesting. When you see a worm, you just think of it as slithering, but what the earthworm is actually doing, munches, so it like bites the dirt in front of it, but then it does this funky thing with its body like that, so it's like making it bigger and then smaller and bigger and smaller, so it's causing the channel, for lack of a better phrase, in the dirt, so it can easily slide through. But it also has these little hair-like structures that I know you don't see, but if you could see one under a microscope, you would see these tiny little bitty things, and that's what helps kind of move it along. Oh, they say bristles here, but it does. It goes poof to make space and then slides on through, and so it does this really cool thing to move through. So that, of course, is going to be friction that it's using. That's also why earthworms are so good for your garden, because it's aerating the ground, letting more in. So the bird, where it says airfoil, it shows the way the wing is shaped. If you have ever seen the front of the wing, it's like that, and then it tapers off. Same way that the wing of an airplane is shaped, because when they were designing the airplanes, they were looking at the wings that worked for the bird. It's because it's making the air flow over it and make it smoother. So again, it's another form of friction. Now we have different types of skeletons. We have the hydrostatic skeleton, which is fluid held under pressure in a closed body compartment. You'll see examples of these. Exoskeleton, X again for exit, so that's the skeleton on the outside. And then the endoskeleton, EN for enter, so endoskeleton is inside the body. Hydroskeleton, or a hydrostatic skeleton, so that's what this is. It's just the hydra filled with water, and that's what holds it in its place. Exoskeleton. This is why when you see this is the old shell, and this is the actual crab. When the crab gets larger, or any kind of creature that has the exoskeleton, in order to grow, they have to shed their old skeleton because it doesn't fit anymore. This is why somebody on an assignment, and I honestly think it might have been a Bio 123 test, they made a comment about loving SpongeBob, and I pointed out that SpongeBob is actually scientifically accurate on a lot of things. This is one of them. There was an episode in SpongeBob where Mr. Krabs is supposed to go to his high school reunion, but he doesn't want to go because he outgrew his shell and he was naked, and then SpongeBob put on his old shell and tried to act like he was Mr. Krab. No, the sponge is not going to put on the shell. But the fact that they showed that when he grew, he had to get rid of his old shell, and then it takes a moment for it to harden up for his new shell, that is completely accurate. That's how an exoskeleton works. This is showing you the exoskeleton of a mollusk. And so that's what this part is. You've seen these kinds of shells if you've gone to a beach store. And then the sea urchin. This is what the actual sea urchin looks like. This is its skeleton, so it's got the little parts that pop off of it. And then we get to our endoskeletons. When you look at a frog, you do not see the skeleton because it's on the inside. This is showing you that it has bones and cartilage. And as I'm assuming you know, we also have bones and cartilage. So, what are the advantages and disadvantages of an exoskeleton as compared with an endoskeleton? Well, exo offers a lot more protection to all the body parts because it's on the outside. However, you are very vulnerable every time you grow because you have to get rid of everything for that amount of time. So now we're to the vertebrate skeleton. Vertebrate skeletons consist of an axial skeleton and the appendicular skeleton. So, axial, that is the center. So just think that. Because if you think of, if you think of the axle of your wheel, and then I realize a lot of people probably don't think of the axle of their wheel. But if you think of the axle, the axis, that is the center, and then everything else comes around it. So for us, axial skeleton, it's our center. So our skull, our vertebrate, and our ribs. That is our main part. That's the axial skeleton. Everything else branches off of that. So that would be the appendicular skeleton. If you have a hard time remembering that, think of in a book, all the extra stuff is in the appendix. So appen for appendicular. I don't know if that helps, but that's how I remembered it. So there we go. Here is a human skeleton. So here you go. For those of you who will be taking anatomy and physiology, my nursing people, start memorizing this because you're going to need to know this and much, much, much more. There you go. So we have our skull, and the only part of our skull that is actually a moving part is this. This is the mandible. I don't think we have a picture later of the skull part, but the skull consists of a whole lot of bones. Then we have the clavicle, which is this. Sternum, ribs, scapula. Pectoral girdle is this whole area. It's what's coming off of the ribs. Pectoral, that means up here because this is our pecs. And then our hips is going to be the pelvic girdle. So pectoral girdle up here. Humerus is this part of your arm. So always remember the funny bone is at the end of your humerus, because funny, haha, and humorous, haha. But so that's the humerus. Then you have the radius and the ulna. Those are the two bones on this part of your arm, which we have looked at pictures of at the beginning of this quarter when we were comparing the bat wing, the human hand, the cat paw, that kind of thing. Then we have carpals and metacarpals and phalanges. Phalanges are your fingers and your toes. Then we get to the vertebrae. This is our backbone. Then pelvic girdle, which is our hips. Femur. It's going to tell you in most of the movies where you see them walking around with a bone as a weapon, it's normally the femur. Patella is your kneecap. This is what I have issues with. Tibia and fibula. And then tarsals. So instead of carpals in their feet, it's tarsals, metatarsals, and then phalanges. This is a handy thing to work on learning. And trust me, A and P, you go in way more depth than that. All right. So we're going to look at the backbone. We have 7 in our neck, 7 cervical vertebrae, which I covered at the beginning of this quarter when I was talking about a giraffe, because they also have exactly 7. It's just theirs is a lot larger than ours. Then we have 12 thoracic vertebrae. Thoracic, as in the thorax. We covered that when we first started talking about body plans. And then 5 lumbar. That's our lower back. So the lumbar vertebrae. And then the sacrum and the coccyx. The coccyx is your tailbone. So there you go. In between each of these, we have the intervertebral discs. If you look at the model we have in class, it shows these little things in there. This is where if somebody says that they have a pinched disc or a ruptured disc or any of that, that's what they're talking about. This is the little cushion in between each of the bones. Just showing you different gene expression during development for this Hox gene. You notice that snake, huge long thoracic vertebrae, a lot of them, but in the chicken it's a teeny tiny amount. That's all this is showing. It's just comparison. Bones, complex organs consisting of several kinds of moist living tissues. We talked about this a little bit already when we were talking some about blood. Shaft of the long bone made of compact bone surrounding a central cavity that contains yellow bone marrow. The yellow bone marrow mostly stores fat. The spongy bone has red bone marrow, and that is where all of the blood cells are made. And then cartilage at the ends of the bones and cushions of the joint, and also things like your ears. So that's cartilage. Showing you the structure of your arm bone, humerus. And it's showing that all of it is in there. So again, the yellow bone marrow, that's fat. The red bone marrow is what is making all of the blood, red and white blood cells. So anyone who has ever eaten bone marrow, chances are that's the part you are sucking out. It just, ugh. When I learned people ate that, it really freaked me out. Anyway, what causes the colors of yellow and red bone marrow? Well, there you go. Stored fat in the developing red blood cells. It's kind of self-explanatory. Now, bones are constantly subjected to stress every day. Every single thing you do, it's putting stress on your bones. Some puts more stress than others. Bone cells continue to replace and repair bone throughout your entire life, and osteoporosis is when it's not actually replacing as well. So osteoporosis is a bone disease that is characterized by weak, porous bones and occurs when bone destruction exceeds the replacement. So again, osteoporosis is when your bone is not repairing like it used to. Examples, we have the healthy bone right here, looks nice and healthy. This is the not healthy. So this is nice, normal, spongy bone tissue. This is osteoporosis. So notice a lot more pits and it's falling apart basically. So when you're little, that's your strongest bone mass because, oh my gosh, if you've ever noticed, a toddler practically bounces. Everything's repairing itself because they know it needs to. Then you're getting up here and you're getting close to your 30s and you still heal pretty quick. Then you're not healing quite as well. Then between early 30s to the 50s is normally when you're getting less and less bone mass. This is the danger zone that if you have not started taking care of yourself properly, you really, really need to. For women, right around here is menopause, and then that is when it plummets. This is also why, if you notice, they're showing you here, it shows both sexes. Here you see woman and man, and then you notice the woman shrinks a lot more than the man. More women than men tend to shrink as they get older. It's not that they're actually shrinking, it's that there are thousands upon thousands of teeny tiny fractures in the bone, and the bone is not repairing itself, and so you just look like you're shrinking. That's why you see a lot more of the little itty bitty old woman than you do the little old man. It hits women more right here because of menopause and the whole change in the way the body's done, and a lot of people just don't consume enough protein, and you need the protein to help replace the cells. How do exercise and adequate calcium intake help prevent osteoporosis? Because the bone tissue responds to the stress of exercise. The more you tend to move, the more you work out, the more you exercise, and when I say work out, I don't mean horribly stressful, I mean just in general. The more you do, the more your bone goes, oh, I need to make repairs. If you're just, then it's not doing nearly as much. So if you know someone who breaks a bone, like say if somebody breaks their foot and they're wearing a cast, so they're not walking on their foot, they're like on the little scooter thing, or they're using crutches, and then they finally can start placing their foot on the ground, but then it's in that boot. That foot is more likely to break again quickly than their other foot, because that bone has not been worked, and so that bone gets thinner and more brittle, and you have to work it back up. It's also why you don't just lay there. It's also why when people are stuck in the bed, they tend to get more unhealthy, and things don't repair as well, because you need the movement to keep it going. So joints allow limited movement of the bones. Well, you have to have something that holds them together, and those are going to be your joints. So we have connective tissue called ligaments that will hold together the bones of movable joints. So the ligaments are bone to bone. I tell you this because we're also going to talk about tendons, which is muscle to bone. Ligaments, bone to bone. So we have a ball and socket joint, as we normally see in our shoulders. That means it's like a poop pops in there. That's what makes it where you can do movements like this. You can curl, 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 curl. Then we have a hinge joint. Hinge joint, like your arm or your knee. It only bends one direction. Think of like when you open or close a door. Door opens and closes on hinges, hinge joint. And then you have the pivot joint. This is the weirdest one to think about, but if you've ever noticed, and I totally recommend you do it, put your hand here, feel the two bones in the forearm, and as you turn your arm, you will feel that those bones actually rotate. So that is your pivot joint. First time I realized there were two bones in my forearm, it really freaked me out, and then I couldn't get past doing things like this where I could actually feel a movement. I don't know. I just find that part fascinating. Okay. Already talked about the ball and socket, hinge, and pivot. I showed you examples. So checkpoint question. Where we have ball and socket joints, horses have hinge joints. Does this affect the movements they can perform? So does a horse move its its legs the same way we move our arms and our legs? Can they move it the same? No, they can't move it the same. So their shoulders and hips work like our arms, like our elbows and knees. So there you go. That's why they're less flexible. How weird would that be, though, to see a horse jump up and just do this? That would be kind of freaky. That would mean the horse could come up and just go smack, which would be interesting. Anyway, muscle contraction and movement. Muscles and bones interact to produce movement. So here you have tendons. Muscles are connected to bones by tendons. Bone to bones are ligaments. Muscles to bones are tendons. Gets very confusing. A lot of people get them mixed up. The way I am going to tell you to remember it is look at how many letters are in the word muscles, how many letters are in the words tendons. It matches the number of letters. That is honestly how I always remembered this. Muscles to bones because of tendons, bones to bones because ligaments. So there you go. Here, where it says they can only contract, what this means is you can only pull. So we might have a picture about this, but I'm going to talk about it right now. If you feel your arms, when you pull this way, you feel the bicep. Your bicep is what's pulling here. To strengthen your arm, this is not going to push. This muscle has to pull. So this pulls, then this one pulls. It's why when you're working out, you know, if you do the bicep curls, that's to work these, but tricep curls are different. And one of the best ways to work your triceps is to put your arm up here, line it up with your ear, and then go back here. If you do this, you will feel this muscle right here work. Now, why is that important? Well, if you've ever been afraid of having that bye-bye arm, you know, you see the person who waves and then this part just keeps on waving. That is your tricep. So again, that's the good way to work on that. So just so you know. Alrighty. This is what's showing you where when you pull, the bicep is contracted. And then when you bring it back out, it's the tricep that contracts. So they're paired in order to make it happen. Again, the muscle only pulls, it never pushes, it only pulls. When exercising to strengthen muscles, why is it important to impose resistance while both flexing and extending? It's because you're working both parts. That's all there is to it. That's the antagonistic pairs. So, if anybody ever went to the gym, did weights or anything, you've got those couple of category of people. You've got that person who is just wanting everyone to see how much they lift, and like, and then they drop it, and you're thunk, 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 thunk. Or if they're on the machine, that's the thing, you keep hearing clank, clank. That is not the proper way to actually work your muscles. That is only working this part, not this part. To properly exercise the muscle group, you have to control the up movement and control the down movement. That's the reason why, if anyone's ever been like, I can lift more than that, but then you're like, if you're trying to control the downward movement, it's because you're strengthening a different batch of muscles. So for anyone who works out, remember, control up, control down. Don't just do that where you're doing that kind of thing. Oh my gosh, it drives me nuts when people use more of a centrifugal force, basically. They're using more force to toss it up than actually lift. Don't worry about what other people think. Do what's actually going to give you the benefit of what you're trying to do. Now, muscle fibers, myofibrils, thick filaments, which are primarily containing myosin, and thin filaments, primarily containing actin. And then sarcomeres, which repeating groups of thick and thin filaments are the contractile units. Contractile units, as in there's what helping pull. So, the two most abundant proteins of myofibril are actin and myosin. There you go. According to the sliding filament model of muscle contraction, the myosin heads of the thick filaments hydrolyze energy and extend to high energy states. Then the heads are going to attach to the binding sides of the actin, and then that continues on. Which region of the sarcomere becomes shorter during contraction of a muscle? There you go. The light bands shorten. Motor neuron, motor, that's what's causing everything to happen. That's the one doing the work. So it carries the action potential to the muscle cell, which causes the thing to contract. Neurotransmitter, acetylcholine, we've talked about this before, released at the synaptic terminal, triggers the action potential. We covered this on Monday, so I'm going to skip over that. How does the endoplasmic reticulum help regulate muscle contraction? I like to think of this as a throwback question because it was Bio 123, Chapter 4, I believe, where you had to learn about the endoplasmic reticulum when you had to learn about the cell. Well, this is how. It regulates the cytoplasmic concentration of the calcium ion, so it decides what's going in and out. And I talked about, again, in class on Monday, about how calcium helps with the contractions. Now, Bio 123, I mentioned fast twitch and slow twitch muscles. So I'm going to take a moment to remind you about this because this is going to be your lecture assignment right here. Fast twitch, slow twitch. You have to remind me about this. Now, in this class, I know I have a couple of people who did not have me for Bio 123, so I'm doing a recap. When I first talked about fast twitch and slow twitch muscles, I said the fast twitch is for the quick burst of activity, hence fast, and slow twitch is prolonged activity. Fast twitch is the white muscle, slow twitch is the red muscle. Fast twitch, think of a chicken or a turkey. Fast twitch, breast, wings, that's your white meat. That's your white muscle, because chickens and turkeys do not fly south for winter. When you see a chicken or a turkey fly, it's a quick little, it's just a quick little burst. That is the white muscle mass. Yeah, I mean, it also has to do, there's no myoglobin and stuff like that. There's other stuff. But the only part I want you to really worry about is fast twitch, white, quick burst of activity, and that I say to remember it by looking at a chicken or a turkey. That's your white meat. Conversely, dark meat, red muscle, dark meat, legs and thighs. Chickens and turkeys walk around all the time. That is what the slow twitch muscle is. So that is your hybrid assignment for this week. Tell me about the fast twitch and slow twitch muscles and what I told you to remember. There you go. Now, brings us back to the aerobic respiration. Aerobic and anaerobic. Anaerobic you also call fermentation. Aerobic respiration contains constant supply of sugar and oxygen, provides most of the energy used to power muscle moving during exercise. Anaerobic starts producing the energy faster, but produces less. And again, we covered aerobic and anaerobic in Bio 1, 2, 3. Now, this is just showing you sources for ATP, energy for athletic activities. So take a good look at this. And then that should tell you whether this is using the fast twitch or the slow twitch muscles. So compare the processes of aerobic respiration and lactic acid fermentation with regard to the substances required, ATP output, and speed. Aerobic respiration requires glucose and oxygen. Remember, aerobic, oxygen. Fermentation requires glucose, not oxygen, because fermentation is the same as anaerobic. As soon as you add the an to the aerobic, that means no oxygen present. And then aerobic respiration produces many more ATP molecules than fermentation, but fermentation produces them faster. There you go. All right, here's where it's going to mention fast twitch, slow twitch fibers. So fast twitch, rapid and powerful, but they fatigue quickly. So again, fast twitch muscles. Slow twitch, sustain repeated contractions, slow to fatigue, but their contractions are less forceful. Prolonged activity. Walk, 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 walk. And a third fiber type, which has some characteristics of both slow and fast fibers, is also abundant in human muscle. That's why it's not part of my example for the fast twitch and slow twitch muscles, because I'm talking about chickens. So, it's talking about how it works, how it breaks it down. Again, slow fibers versus fast fibers. How it happens, we've got aerobic, we've got anaerobic, myoglobin, I mentioned this a moment ago. Lots of myoglobin in the slow twitch, very little in the fast twitch. That kind of thing. This is also why the flavor is different in the white versus the dark. And genotype frequency, not going to worry about it. What observation about the protein actin 3, I should say alpha actin 3, led researchers to hypothesize that actin 3 genotype might affect an athlete's success in power events? Well, only found in fast twitch muscles. So there you go. And that, I was about to say that, my dear. That's it. We're done. That's done with chapter, what was that, 29 and 30. You have exam 2 this week. Make sure to be ready for it, and hopefully you'll do okay. Look back over your quizzes. There's a couple of questions that might be on there from these presentations, and if you just simply go to the very end of these lecture videos to figure out what the lecture assignment is, I mentioned it earlier in this video. So if you just jump to the very end to find it, you're going to have to go backwards and find it. So there you go. I guess see you Monday. Peace out.